Hello, everyone, and welcome to the Genetics Podcast. I'm excited to be back today with one of our guest favorites, Dr. Veera Rajagopal, who's a drug discovery scientist at Regeneron. You will recognize his voice if you listen to our end of the year recap episode in 2023 or in 2022. This is one of the episodes that we get really great feedback on. People love to hear some of the latest and greatest in the world of genetics and precision medicine. And as you all have heard me say a couple of times, I just think Veera has an incredible mind for finding these really interesting stories and making sense of them. So we thought we'd try to do this a little bit more frequently because obviously the news is always coming out and there's always interesting stuff being published. So thank you, Veera, for taking the time to do this again.
Thank you, Patrick, for inviting me again for the third time.
Yes, and many more to come. I'll just say, if you're on Twitter or X, I guess we have to call it now, you should follow at Dr. Veera. We'll put these links in the show notes as well, and also follow the GWAS Stories Substack. If you want to see any of these stories in written form, then a lot of the stories we're going to go through have actually already been written about by Veera over the past couple months. We're going to cover some interesting findings and some broader topics that these sit under that have come out over in the last couple months. And the first one we're going to start on is actually some really interesting stories in the world of non-coding variation. Longtime listeners of the podcast will know that this, way back in the day, was what I did my focus in my PhD on. So it's a topic I'm really interested in particular non-coding variants in rare disease, which a couple of these stories are about. And one of them has been, this really kind of famous non-coding enhancer that causes polydactyl, which is having an extra digit on the hand. And this locus is something that's been studied by practitioners and interested people in the non-coding genome for a long time. But there's been some really interesting work that's come recently that I think sheds some interesting light on some of the fundamental mechanisms of how transcription factor binding works. So Veera, I'd love if you could talk a little bit about the finding and also what struck you about this as being interesting and worth writing about and talking about.
So I was like very interested in this paper because I have the special interest on the emerging new discoveries in the non-coding space. I think we had topics related to non-coding variants in both of our yearly episodes. So this is still we are just beginning to understand how disrupting the non-coding part of the genome, which is 98% of the genome, actually leads to human diseases and traits. And there's like large scale efforts right at the moment. The UK Biobank whole genome sequencing has been released, and people are, I'm sure, diving deep into that to identify some of the low-hanging fruits of non-coding variants causing diseases. But generally, so far, I have seen as some of the best insights, be it non-coding or coding, always come from patients with Mendelian highly penetrant conditions. And, it's less isolated cases, but profound insights. And we had so many examples on that. So we saw about how a non-coding variant that can disrupt the tissue-specific expression for hexokinase 1, causing hyperinsulinism. And also we saw like non-coding deletions that can have effects equivalent to deleting the gene itself for hyperinsulinism. FOXA1 example in the last episode. And so this time for us, there's a couple of examples to talk about. There's like gain of function effects. That is also very interesting. And so here it's not really a genetic discovery, but it is some deep dive into the mechanisms of how this non-coding variants affects. And so this is from Emma Farley, who's from University of California, and her team actually is an expert in studying the mechanistic aspects of non-coding variants, particularly on enhancers, transcription factor binding, how they are conserved across species, and how they actually harmonize this developmental trajectories, etc. So I think so here they actually study a specific enhancer called ZRS enhancer, which is very well studied and is known. It is involved in the development, particularly one of its target genes, SHH gene, is involved in limb development. And so mutations in this enhancer causes polydactyly and supernumerary fingers, and it's known for a long time. But the special interest for, I think, for this team to look into this is come from their own work early, where they actually, I think it's a science paper published in 2015, where they found some remarkable, logic behind transcription factor binding and how they actually affect the development. I think it's the first time they actually found it. So they studied the sea creature, the sea squids, which is one of the closest living relatives of vertebrate species. I think it's like separated from vertebrates maybe like 200 million years ago. And so there is a lot of interesting commonality between these two. So they actually studied a particular enhancer that is like just 68 base pairs and that is involved in the development. And they did some MPRA assays to, find all combinations of this enhancer motifs to see how it actually affect the transcription factor, known transcription factor that binds to this enhancer. And the fascinating discovery is like often when we think about transcription factor binding to the targets, it's like we always think about the protein binding with very high affinity, right? So even if you're given a bioinformatics task or anything to say find the regions within a sequence where a transcription factor, bind, which region is important, like we tend to think like wherever the region, the transcription factor actually binds with very high affinity, right? So here they just find the opposite, like they when they actually did this massively parallel reporter assays and all the combinations. So what is wild type? Wild type is something, we know it is very specific for this development function. So you would assume the wild type to have the perfect binding with the transcription factor and having the most highest affinity. But they found that the wild type sequences are the ones that had the least affinity with the transcription factors. And if you actually scramble it, you can actually push the affinity to very high, so highest affinity. So compared to the highest affinity, the wild types one actually have like very subtle, like probably 15%, 20%. And so this is first time they actually finding this kind of a logic, transcription factor finding logic, where suboptimal fine binding is what is actually driving the tissue specific expression. And there is also like a redundancy in the binding motifs in the enhancer regions, where each of the motifs themselves are not like, indispensable. They are redundant, but together they become dispensable. I think it's a very unique evolutionary design to prevent against maybe loss of function effects or anything. And so the unique aspect of this is comes with a like a um, disadvantage of being susceptible to gain of function variants. And so if there are naturally occurring mutations in this enhancer in this C creature actually can causes developmental anomalies. Sometimes it can be as severe as having a two beating hearts. So their work, they actually thought, okay, they wanted to see if this phenomenon is also seen in humans, and they chose this ZRS enhancer associated with polydactyly. So it is, I think, so 800 base pair enhancer, very well studied. And so they actually, found all the, curated all the known polydactyly mutations. And they started looking into how the transcription factor binding, profile works here. Is it very similar to what they actually learned from the CNO species, right? So, and so I think like they found that the transcription factor binding are very similar to what they know is like the wild type sequence actually have a very, like low affinity binding, and then the mutation associated, like binding motifs actually has high affinity. And the interesting part is that just a very, very subtle change in the affinity actually, causes ectopic expression in the lute development and causes polydactyly. And this is also very important because we always look for big fold differences between when we do this assays, right? So whenever we want to see screen for disease associated variants, we actually see what causes the maximum fold difference in binding affinity or whatever, right? So sometimes the ones that matter is actually the ones that cause very small changes. So this lot of findings here actually, like inspiring scientists to rethink how we actually do this kind of assays to identify or to understand non-coding mutations. So I think it's very interesting. So they developed, like they also did massively parallel reporter assay to focusing on a very, like one particular binding site motif. And then they find that, combinations that can increase the affinity very high. And they also see that as the affinity increases, the penetrance of the disease also increases. And so they created synthetic SNVs where you can increase the affinity, and then they see when you actually knock into the mice, actually it causes more, supernumerary fingers, affecting forelimb and also hind limb. So I think it's like one of the example to remind us that what we know so far about non-coding
Mm.
variants mechanisms is, it's just very, very superficial. There is so much more to understand. And we should continue to study all this kind of highly Mendelian conditions. And I think that is where really a lot of insights continues to come. Um,
Yeah, I agree. And I think the big challenge that we have as a field is if we think about the coding regions, we understand the triplet code and we know which changes cause which amino acid changes. We know which changes are going to cause a premature stop and break the protein. And so we've known that for a long time. And there are some exceptions, and sometimes we learn surprising things. But for the most part, we're really good at predicting what a DNA change is going to do to a protein. But we're so far from that in the non-coding region. We just don't really even understand the grammar. We can read it, but we don't, we except in a couple of these really well-studied loci, we're still seems like we're pretty far from understanding what that universal language is, which I think is really interesting, but it's also a big challenge. And the reason I follow this field is obviously I spend a lot of time working in it, but also I think for the rare Mendelian diseases that have causes of the non-coding genome, we need to understand this grammar if we're going to diagnose those patients. But also, I suspect there's probably something in the common disease world where where you spend a lot of time from a drug discovery perspective. If we know that a lot of the signals in the non-coding region, but it's sort of an issue that we don't really know how it works, right? We often don't even know the driver mutation or driver variant underneath it. So I was curious whether you have a thought on how solving this problem or getting closer to solving it might help in common disease, common variant world where these are not massive impact changes, but they're more subtle. But I would imagine they probably still sit within this wider framework you described of we've got suboptimal affinity binding, and so these changes are either subtly changing the expression of a gene or they're subtly changing what point in time or what location in a tissue a certain gene is flipped on and off. And I'm just curious whether you have a thought on how better understanding this might help us to do better drug discovery, for example, or target discovery.
Yeah, I think so the one of the biggest value of this like large resources that we get from the UK Biobank is to apply our knowledge that we learn from these extreme, like rare diseases, right? So the rare diseases and common diseases, they always lie in a spectrum. So just blindfold going and searching for, important findings in a non-coding space, it's a very big space. So it's impossible to pinpoint which ones are important, what are the mechanisms. So I think we need some kind of help to narrow down our search space. And
Mm.
that can come from rare diseases, for example. So this paper actually might have inspired other people to actually go specifically look for subtle changes in affinities and focusing on developmental enhancers. So, so if there is like, so what other enhancers? I think one of the extension of the additional work they did in this paper, where they also looked into the other enhancers other than ZRS, and they also find that probably the same logic actually helps there. So probably, so just piggybacking on this mechanism and then going back and searching through, in hundreds of thousands of individuals, and you have other layers of omics data, you can actually try to find, more similar findings there, where with mild effects compared to, severe effects. And so I think that is the major, uh, like, towards what the people are actually moving towards. I think, like, I always say that, we have this UK biobank, but the real value comes when you actually bring in annotations, developmental stage specific annotations, tissue specific annotations, not just simply, databases, but more insights on what specifically you're looking for, what kind of assays you want to do. So once you have those, then when you combine with the UK biobank or emerging whole genome sequence, I think you'll find a lot of more interesting things. So just plain blindfold search, like, it's I think it's probably will yield some of the low hanging fruits, but not as many as people are hoping for. Particularly, it's more challenging in the drug discovery space. I think that is why a lot of people are still reluctant to move from exome to genome, in terms of, using human genetics to drug discovery.
Yeah, definitely. This is a topic we'll cover, I think, in a future discussion around this dichotomy of genomes and exomes, and at what point we may transition clinically and in a research, or whether we're getting enough from the exome. I know you've the Regeneron has published a preprint previously on this topic, for example. I wanted to just also switch to the second paper here, which is equally interesting and along the same lines of really focused on rare, high impact, Mendelian disease cases. And I also found this paper that you shared super interesting, because I spent 3 or 4 years of my PhD hunting for non-coding causes of severe neurodevelopmental disorders, and we found we didn't really find any smoking guns. We found a lot of statistical evidence that the non-coding genome was involved and was contributing in a significant way to these rare diseases, but we didn't find a great smoking gun like this group has found with a really interesting deletion of a non-coding element. So maybe you can talk a little bit about this, and then I'm also happy to talk about why I think these things are so hard to find in general, why the examples that we find like this are amazing, because they come so rarely.
Yeah. So this paper, again, it's also a gain of function variant. So like I mentioned before, that we really, one of the major goals of, extending the analysis from coding region to non-coding region to identify, mutations or carriers who have very similar profile as having a loss of function or gain of function. I think for loss of function, at least there are, handful of examples. For gain of function, I think it's very few examples that we have that comes from the non-coding region. So this is one very, very interesting example. I think it's from the paper is from research team from Broad, MIT, Harvard, and also Northwestern University. There is a fantastic thread from the first author, Dr. Vijay Ganesh. I think he's a neurologist. He has put in the Twitter. So we should maybe link that thread there. I think it's beautiful storytelling there. So the story starts with this one child, Emma, I think it's the child's name, and their parents actually. So the couple is trying to identify a genetic diagnosis for their child. So they noticed that the child had neurodevelopmental symptoms in after 1 month or 2 months old, and they were trying to identify what is the cause. So I think they did exome sequencing. They couldn't find anything, and then they also went for genome sequencing. And in this journey, they ended up at one, some specific set of variants, and one particular variant is a 5' UTR variant near CHD2. So CHD2 is a very well known neurodevelopmental gene, but most of the reported, NDDs are caused by loss of function variants. But still, since this de novo mutation is located very close to it, I think that's how they started digging into this. Then they also did RNA sequencing, and in the RNA sequencing, they found that actually the CHD2 expression is increased. Like the expectation would be that, somehow it is causing the CHD2 to decrease because it is a haploinsufficient gene. So they saw this opposite finding, and then they went back to see, okay, so probably this gene is also intolerant to increase in expression, and that is true based on recent databases where we can actually look at what is called as triplo sensitivity. And but still the mutation is not very convincing. It's a not it's located in a not a conserved region, and though it's cannot cause such a big change in the expression. And so they continued to investigate. I think they did short read as well as long read sequencing, and that actually led them to a 22 kb deletion just a little bit away from the de novo region, and that actually deleted a long non-coding RNA. The name of the lncRNA is called chaser. And so it is at the same time there was a parallel publication where they actually also uh, landed on the same finding, where they show that if you actually delete this long non-coding RNA in a mice, you get similar, encephalopathy related features. So everything aligned. And so the interesting thing is like this actually acts very specifically in cysts. So it affects only it controls only the expression of CHD2 only in its own chromosome. And they actually see that in the allele specific analysis in the RNA sequencing. And so then they wondered how many of such undiagnosed cases in the database can be explained by mutation that actually causes loss of function of the long coding or this lncRNA. And they looked into this matchmaker program, and they identified two more patients actually had different kinds of mutations, but again it's kind of hitting the same lncRNA, causing haploinsufficiency of this lncRNA, causing very similar neurodevelopmental this syndrome as they see in this patient Emma. So it's very interesting because this is, probably there is some therapeutic opportunity here, because always it is easy to treat, design a therapeutic drug to inhibit a gene or, like a protein rather than increasing the expression. And so when you have like this here, so probably when the disease is because of gain of function of the CHD2, because of the the long non-coding RNA, you can actually probably inhibit the CHD2 itself. Or if in the other condition where you actually where you have the NDD because of loss of function of CHD2, you can actually inhibit the long non-coding RNA to increase the CHD2. So you can fine tune it.
Yeah.
And this mechanism is being explored in the Angelman syndrome. I don't know if we had an opportunity to talk about this. A very interesting, condition. There's like two condition, Angelman, Prader-Willi, both caused by deletions in a specific region in long arm of the chromosome 15. It's one of the oldest known CNVs, and there is it's a constellation of syndromes, and still I don't think there is any clarity on what gene causes what syndrome. But at least for the Angelman, which is actually caused by the genes that are maternally imprinted, so if only the genes that are maternally expressed, it get deleted, it causes this Angelman syndrome. And this very specific gene, UB38, so ubiquitin ligase, actually is the main one that underlying the Angelman syndrome. So one of the major discoveries there is that they identified the mechanism of imprinting in the paternal allele. So there is this long non-coding RNA which actually goes and completely imprints prevents the expression of UB3A in the paternal allele. So one of the therapeutic design right now is to release this imprinting, this inhibition by inhibiting the long non-coding RNA in the paternal allele. So the loss of function, the loss of UB3A in the maternal allele can be compensated by increasing, activating the gene in the paternal allele. And it has been, so far successful. I think my studies showed promising results, and right now, I think we will be seeing some readouts from the clinical trials of this. So this kind of reminded me that CHD2 reminded me of this one. And so it's kind of make you wonder how many of such long-coding RNAs are there in the human genome, and how you can exploit it to, treat diseases caused by haploinsufficiency, for which we didn't have a solution actually to increase the gene expression. So it's like super interesting paper overall.
Yeah, it is. And I think part of what makes discovering these so difficult in the non-coding regions is, I think, related to this concept you described of haploinsufficiency on one side and triplo sensitivity on the other. But it's kind of a spectrum where, for those who aren't familiar with the terms, haploinsufficiency means that if you have just one functional copy of the gene, you have, typically problems, some kind of syndrome. It's having one copy is not sufficient for full functioning. And on the other side, what you described around triplo sensitivity is having three copies of the gene does cause problems. And so there's space in between when we think about subtle changes to enhancers, non-coding RNAs that mean some genes are going to be really tightly regulated and may only be tolerated between 90 and 110% of the right expression level, whereas others we know, housekeeping genes, for example, need to be tightly controlled, but other genes can vary widely and have zero copies or one copy. And I think what makes this also tricky is this is probably a tissue dependent thing as well. Some genes are tissue, it can vary widely in some tissues, but needs to be very tightly controlled in others. And so I think this is a multifactorial problem to
Also
figure out.
developmental stage specific,
Yes.
Yeah.
Yes, that's right. Tissue, time, developmental stage, and so it's quite also some of what makes this fundamentally difficult is getting access to tissues during these developmental time points is almost impossible, right? Because you're talking about understanding healthy fetal development, and getting access to healthy fetal tissues during development is just not something we can do. So I think there's a lot of practical challenges with. There's almost a black box of we have a very difficult way of knowing what does dosage of these particular genes look like in humans at particular developmental time points. We can only study it in mice and other organisms, right? We're ethically unable to really understand what's in that box for obvious reasons.
Yeah, definitely. So one of the other thing that I really hope that non-coding variants will help us to is, one of the things that coding variants often cannot tell is like, differentiate between essentiality of a gene based on, prenatal versus postnatal. So one of your previous episodes from Alnylam's, VP Paul Nioy, so actually was telling about this angiotensinogen siRNA, right? So basically, it is a developmental gene because if you're born with some loss of function, actually causes kidney problems, and we don't have human genetic evidence, but still it is safe to inhibit it, using siRNA and to replace the small molecule for antihypertensives. But that safety thing that cannot come from a human genetics perspective. But probably, there are examples where for example, HK1 hexokinase, or there's non-coding mutations that can inhibit the gene expression within specific developmental stage. So if we find non-coding mutations that can actually inhibit the gene, for example, angiotensinogen, if you take that example, postnatally but not prenatally, and it has a protective effect on the hypertension or something. So such examples, I don't know, I'm just curious if we'll be able to find such examples that can actually differentiate, have very developmental stage specific, tissue specific knockdown or knockout that gives insights into drug development. That's one thing that I'm excited about, but I don't know how, if you'll be able to find such examples.
Yeah, and interesting. We'll keep an eye out for them. The third and final paper in this non-coding theme is I think you put in our notes that you thought this might be the most interesting of the bunch, and I do think it's really interesting as well. It's we're starting to translate out of Mendelian world and into more complex. I don't know if I'd necessarily say common disease, but common in some populations. It's a really interesting story here. I'm not going to spoil the details. I'd love for you to tee it up and talk a little bit about this Brugada syndrome story that is really an interesting one.
Yeah, so yeah, like I thought this is probably the most interesting one. And this was actually the preprint came out in December, but we couldn't get that in our year roundup. So I added that to our, in the January roundup that I wrote in the Substack. So, but yeah, definitely super interesting. We cannot miss this in our podcast roundup. So The one reason why I find it interesting is like, it has some commonality with other. So basically, this is Brugada syndrome, just caused by this, arrhythmia related condition. Heart conduction defect that causes sudden cardiac death. And so these causes for sudden cardiac death, particularly like the ventricular fibrillation arrhythmias, actually have a very fascinating history. If you think about how people dealt with these kind of conditions in the past, like uh, a person completely healthy, no signs, signs of health disease or anything, and then suddenly they die, out of the blue. And so this book, Genome Odyssey from Yuan, actually, it has
Yeah.
like a very interesting story on this Jervell-Lange syndrome. It's a long QT syndrome. It goes all the way back to the 1850s, where there is like this book from a headmaster of a deaf school where he documented specific experience where the small girl who actually stole something, and he wanted to punish her by like calling out her name in front of the school assembly, and then the moment he did that, actually she collapsed and then she died on the spot. And then later he learned that the whole family, it's not the first time someone is like just falling suddenly and collapsing and dying. So then 100 years later, I think the Norwegian scientists found it, and the story goes like that. So very similarly, there is like one of the other common cause of long QTs, like sorry, ventricular fibrillation, sudden cardiac death, is this Brugada syndrome. And this story starts, it goes back to the 1980s. And so this was like first time, the CDC is actually came to know that alarmingly increased rise in the sudden cardiac deaths, particularly in the Southeast Asian immigrant workers. And so, they did a lot of investigation, and, they couldn't find anything. So the clinical features is like very fascinating. So almost everyone are healthy young males. Most of them are males and no previous history of cardiac disease or anything, and everyone died, like they went to bed normally, and then they just died in between 10 p.m. to 8 a.m. And there is no like signs of struggle or anything, and it's just they get sudden cardiac arrest and then they die. And then that's how they realize that there is something that is really common in Southeast Asian men. And so that's when they actually started. And then when this news came, there is also other parallel reports in Singapore, where workers from Thailand also was like dying very often because of sudden cardiac death. And this kind of inspired, three cardiologist researchers, a team of cardiologist researchers from the Thailand Bangkok Heart Hospital, so who actually started studying this 30 years ago. And it was at the same time, like, we came to the Brugada syndrome actually was born in 1990s or somewhere. So where is, Brugada brothers, they are like scientists in Barcelona, cardiologists, who actually he was studying idiopathic ventricular fibrillation syndromes, where you have this ventricular fibrillation causing sudden death, where you don't have like a specific cause. And so they identified a subset of people with very characteristic ECG pattern. I think the ST elevation in one of the a couple of the leads in the ECG, and and sometimes it won't be found, but you need to give some kind of blockers, channel blockers to evoke it. But it's a very characteristic, and that describes a subset of this idiopathic ventricular fibrillation, and that's how it was named after this cardiologist who actually first reported it. But it was almost the same time there was this research happening in Thailand. So these people also, like this is cardiologist Gumpanath and Apichai, and it's very sometimes tricky to pronounce this Indonesian names. Tinkun Lavi Nedmeni. So they actually worked on similar, clinically they were working on the patients, and they published a paper almost the same, just probably a few years or the same, it's a few months maybe on after this Brugada syndrome was published. And so they show that the sudden cardiac death causing condition in the Southeast Asian men is like very similar to what was described by Brugada. And then that came to the realization it's actually Brugada syndrome, and it's extraordinarily common in Southeast Asians. So it's sometimes the statistics can be impressive. Like I think I saw something like 20 to 30 deaths per 100,000 deaths in young men is
Oh,
actually caused by Brugada syndrome in certain regions of the Southeast Asia. It's very common actually cause of mortality there, and it's very well known for a long time. So they have some ancient tales about how this, like, because you just imagine how our people would have seen this, like going to bed and then dying in dreams. I think there's like many interesting tales, like names Lai Tai, I think that's how they call it in and then there's like Pokuri, I think, in Japan. So there's like many different names. It's very well known for a long time. But that's just only in the 1990s they first discovered, okay, this is they discovered what is the disease, and then in a few years down the line they actually found the gene. But surprisingly, even though this was like extremely common in Southeast Asia, the discovery came from Europeans. It's just based on 8 patients, and they specifically looked for mutations in the known ion channels, and they've identified, like in all the patients, some low loss of function variants in SCN5A coding variants. So it turned out, like among European populations, almost 20% of the cases can be explained by low frequency, rare, deleterious coding variants. And it is also, not like a Mendelian kind of disease, not a rare, caused just by rare disease. It's a polygenic disease. There is also GWAS studies that show strikingly strong, signal in the SCN5A, where at least you have like a dozen independent locus, many non-coding regions, but these are common variants, increasing the risk by a little bit. It's from in the polygenic space. So the thing is, like, we know that this Brugada syndrome is extraordinarily common in Southeast Asia, but they never came to find the genetic mutations actually causing it. There were studies focusing on SCN5A. They found one low frequency missense variant, but it explains only a little bit, and then so overall, just only 5% of the cases can be explained by some kind of coding variants. So that kind of, inspired these people to look into the non-coding variants. So there's this team of scientists from Amsterdam University Medical Center, Connie Bezina, I think it's a PI who actually is leading on working on this for a long time. It's the person who actually also found the gene,
mm,
in collaboration with actually the Brugada brothers, who also was part of this gene discovery. So she works with this Bangkok hospital researchers who've been like working on this Brugada syndrome for a long time. They have like, I think, decades of collaboration. And so they started looking into the non-coding regions, whole genome sequencing, and then they found, so recently, like they found this one set of non-coding variants all in high LD, and they found this, like they landed on this causal variant by doing functional experiments. And it turned out this non-coding variant is located within an enhancer that where the cardiac specific transcription factors bind. And this actually reduces the expression of the SCN5A by like active sodium channel activity by like 30%. Single allele, it's almost close to having a loss of function variant. And this is seen in 1 in 25 cases of Brugada syndrome. It's extremely like common compared to other regions, and it seems to be very specific for Southeast Asia, particularly Thailand. And so it's I think it's the very first time a non-coding variant is being discovered to explain the high prevalence of a disease condition in a non-European population. So for a lot of reasons, this is like really super interesting. This is also very similar to the Notch3 mutation that we've been discussing last time for the stroke in
yeah.
the Pakistani population. But here it is a non-coding variant, and it's amazing. It's more fascinating when you actually look into the history, how it and knowledge is being evolved, and it's really a very fascinating literature. Actually, I recently only started looking into it.
Yeah, I'll recommend that everybody goes and reads the January edition of your Substack where you go through the story, and I think you've included quite a few links there. But I agree. I find it fascinating to understand how these things unfolded over time, and also whether there were times in the past where something like this might have been discovered, but it wasn't for whatever reason, right? It's interesting to understand where we're, where there are delays in science because someone missed something at one point. I don't know if that's the case here, but I was one thing I wasn't sure about was whether there was any good like evolutionary explanation why it's so much more common in this population. Did you get that from any of your reading of why it might be so much more common here than or
There is
there
a
somewhere else?
speculation. It's definitely there is it probably not under natural selection, because it's the people die very late in age, right? No more than 40 years or 50 years. But they speculate in the paper saying that, we know APOL1 and all similar stories in African population, where there is a trade-off within protection and disease risk. So probably the increased susceptibility to Brugada syndrome could be a trade-off or something else, that is, like some other beneficial effect that might be. But I don't think like they have any looked into that any, there's no evidence right now. But also it's a rare variant. It's not like a common variant
Yeah.
like Apollo or anything to have that kind of evolutionary explanation. So if even if there is something that is probably, recent selection or something. But yeah, it's interesting to think about. But a lot of times it is always just it's a natural drift.
Right.
It's a random increase in this population with no specific reason.
Yeah, that makes sense. Speaking of drift and populations and what we can learn from non-coding and coding variants, I thought it'd be good for us to finish on a final paper here, which falls under a slightly different topic, which is around what does the future of genome-wide association studies look like? We discussed on a previous episode how there's an amazing paper last year where they did a saturation effectively GWAS of height and felt like the job was done. Basically, we found found everything we're going to find. Obviously, it's not quite the whole story, but we're starting to do more and more GWASs at the millions of person scale. We're getting to scale in many different populations and ancestry, backgrounds and genetic ancestry structures. So I'm curious whether you could talk a little bit about this type 2 diabetes paper and maybe talk a little bit about whether this is the direction that we might see the field go, in particular, starting to break down disease mechanisms and some of the differences that fall along genetic ancestry lines that we're getting from this paper.
Yeah. So this is something, I'm generally not, I don't get that much impressed with GWAS papers with huge sample size, unless there is like really great message that comes out, like a height paper, right? So one of the things that height paper showed us, actually, at least for the quantitative traits, you can reach some of the target goals where people want to reach, like identifying all the possible common risk variants that you can find to explain the full heritability explained by common variants at a genome-wide scale. And then, so that kind of saturation we might never reach for binary traits. I think there are some theoretical papers giving estimates of how much sample size that you would need to reach a
Interesting.
saturation for different diseases, and it's probably will never go reach that point. So but still we see year by year, GWAS sample sizes are really increasing. And one of the major, like the diseases that are very far in the, very first in the race is like type 2 diabetes, coronary artery disease. So these are the, diseases where you have huge consortium, and then the sample sizes just builds up. So one thing that I am personally interested to see, okay, you cannot just keep reiterating the same kind of analysis number of genome-wide significant loci, and like some enrichment, tissue specificity, enrichment, etc., polygenic risk score. But that ultimately there should be some new next generation thinking ideas to come up. I think like this is the approach that I was like that interested me is like not a new one. So what they're actually doing here is like they are clustering the genome wide significant variants based on different disease mechanisms. And this has been done, I think, at least for the past few years, particularly in the type 2 diabetes. So we are talking about, so here there's like two recent GWAS papers came out in the type 2 diabetes. One was published in the Nature, where also they use this kind of clustering the variants. But then we also have another paper published in Nature Medicine. So we will link both in the notes. So this is from Miriam Adler from MGH, Massachusetts General Hospital, Boston. I think her team is like very interested in understanding pathway specific disease risks, and they have actually previously published also clustering the type 2 diabetes risk variants. So in this paper, so both have very similar approach. So they actually use this genome-wide significant variants to cluster them and then study the cluster specific disease downstream analysis. So this is interesting because like one of the thinking in the GWAS is to how do we dissect the heterogeneity, right? For complex diseases like obesity, type 2 diabetes, the causes of heterogeneous. Obesity can cause type 2 diabetes, like some other risks also can cause type 2 diabetes. So all this comes under, will you find disease risk variants for all of these causes? A lot of these causes themselves are heritable. So one way to approach this is to be very careful in how you curate the disease, stratify the cases based on their BMI or family history or in their risk factors, environmental risk factors. But it's incredibly difficult in a scale of what we have today in the number of cases. So the other way is like we are getting enormous power in the genetic discovery. So you can actually address this heterogeneity using the discoveries itself. Just as the sample size gets bigger and bigger, the power becomes higher and higher, then automatically you get clarity in the results, like so when you see that, you can see there's like a set of variants that are pointing you to the beta cell biology. There's a set of variants that are pointing you to the obesity. There's a set of variants that actually pointing you to the behavioral aspect that actually contributes to obesity and appetite pathways, things like that. And that is getting clearer, at least for GWAS, like type 2 diabetes. I think the similar approach also used for NASH. I think there was one GWAS that also showed this pathway specific analysis. And this is like there are at least a couple of things that for me it's very interesting, is because one is stratification, right? So polygenic risk score is being more and more being used in the clinic, right? There was one important paper from all of us where they actually show for 10 common diseases their experience of actually handing over returning results to the patients about their polygenic risk. And it seems that it's now it's getting more normalized to use this. But there are like one obvious issue that we know is that the ancestry differences between the populations. But then there is like other thing is also one of the things that I'm interested in, how like the polygenic risk score is a blind folded approach, right? So you just pool all the variants, so you don't consider pathways or anything. But that is perfect. So if then you stratify them, saying top 1 percentile, bottom 1 percentile. But there are situations where a person might not have a higher polygenic risk score as a globally looking at all the variants, but they have clinically they have a severe disease because they have a set of risk variants that are hitting in the same common
Right.
molecular pathway. So that is the idea behind pathway specific risk score. I think there are many people working on it. Kristen Bernard from Yale University, she is working in this aspect on the schizophrenia genetics. I think I listened one of her presentation last year World Congress, where they show that a person can still, if you stratify the persons based on pathway specific risk score, then you can identify people at very high risk, which you cannot identify using, overall polygenic risk score. So this stratification is really important. And this stratification also goes to, comparing between the ancestries. So this Nature Medicine paper, they have this different clusters. One of the clusters is lipodystrophy, where, it is abnormal distribution of fat. So we know there's very well that, people differ in the way that they store fat. European ancestry individuals actually, often visually they look fat, but sometimes they are healthy because they store the fat in the subcutaneous tissue. And so, but sometimes people from East Asia, from South Asia, they might not visually look fat. They have like very different kind of, like they store the fat inside around the organs.
Right.
And there is a strong genetic, risk factors for how people store their fats. And so this is kind of called lipodystrophy, abnormal distribution of fat. And so they identify this cluster for lipodystrophy, two clusters. So it's a set of variants that are pointing to that. And then they show that these clusters are more important for people from East Asian ancestry, or so also for South Asia, but I don't think like we still have enough samples for South Asia population. So they show, for example, we very well know that BMI cannot be, we don't have a common threshold for BMI to say obesity between the populations. So currently the practice is to set the BMI threshold based on whether you come from, like a South Asian ancestry or European ancestry. And so this difference can be very striking. For example, in the paper, they show what is, the type 2 diabetes risk associated with a 30 cutoff of BMI is equivalent to the risk, it could be equivalent to a cutoff of 24 in East Asia and
Wow.
20 in South Asia. That's going opposite end of the BMI spectrum.
Wow.
So that just kind of highlights how, BMI can be, poor indicator. And so we need other metrics. But what they also show is that when you actually adjust the individual's risk after you adjust their cluster specific risk scores, this differences in the BMI type 2 diabetes risk relationship between the ancestries actually blunt. And
Hmm.
so they kind of envision a future where you decide the BMI cutoff not based on, self-reported ancestry or race or anything, but in an individual genetic makeup, like a precision medicine approach. There's also important, like people often ignore the ancestry differences. I mean, it's very well known that phenotypically there is this big difference, but it's good to see, it's coming up in the genetic, in the space as well. And people are able to find specifically this pathways that matter much more in South Asians compared to Europeans. And this also have implications in drug development. So probably type 2 diabetes focused drugs that addresses lipodystrophy related pathways probably, has a much more impact in the South Asian population than on the European population. So this has implications in how you design clinical trials, how you design drugs, that could be more matter more for particular ancestry. So I think like this approach kind of gives you a glimpse of how things will go in the future. And so type 2 diabetes is a very good example because we have huge cases like a prototype example, but hopefully we can apply this to other conditions like schizophrenia or, neurological conditions. And probably we will be able to finally see some of the biology that we are not able to see so far on this, like, this muddled space of genome-wide significant hits.
Yeah, yeah. I think the point you raised about honing in on people more to have more effect in a really a true precision medicine type approach, you don't even really need to necessarily think about the genetic, the self-reported ancestry, right? You can almost let the genetics speak for itself in this case and say, if it's a lipodystrophy pathway that this particular person is going on, whether they're from Southeast Asia or whether they have European ancestry, the genetics is pointing us towards this being the predominant pathway. I find that really interesting. And it's a very encouraging direction, I think, because it takes what was one of the things that I had always struggled with polygenic risk scores was they seem to be really effective at pointing towards overall risk, and obviously they outperform, and they outperform any small number of GWAS hits, and they also add value to the standard of care in a lot of cases, I think. But they weren't always great at pointing out actually what's the fundamental biology here that's driving this difference. And it seems like, at least for some of these phenotypes where we're at a big enough scale that we can do this, then like you said, the scale starts to unfold this additional layer of detail that helps us understand not only who's at high risk, but why, and also uncover people who may be in the middle of the distribution in the aggregate, but they're really far out on one of these really important endophenotypes, I guess you'd call it, and are therefore actually at higher risk. So I'm glad you flagged this up, because it's a really interesting paper and a broader shift in the field, I think, that I'll keep an eye on. We're out of time. I just wanted to say thank you again. This is one of everybody's favorite episodes. So hopefully people liked it's not 2025 yet. This isn't the wrap up episode for the year. This is just middle of the year check in on some of the latest and greatest that have come out. So Veera, thank you as always for taking the time to be with us.
Thank you very much. Hopefully we'll have more sessions in this year.
Absolutely. Any feedback on this episode or others, you know where to find us. Thank you all so much, and we'll see you next time.